Today, we're sitting down with a real biologist to learn about the origins of life. You guys know the longest word in English? It's pneumono ultramicroscopic silicovolcanoconiosis.
Okay, yeah, that helped me understand this way more honestly.
You know what the word is, but you can't spell it? Come on, man.
And then I left it on and it melted.
Oh my god.
Do you like percolating? That's the first question. One big problem in the origin of life is that we still don't even know what life is. Well, so where did life come from? Nobody knows. Life as a phenomena is not really well explained in physics, and I think it's a piece that physics actually needs. Physicists aren't really acknowledging it, but there's important implications, I believe, to the origin of life and to the origin of consciousness that that have a lot to do with like information or information theory and entropy. And these are big open questions in physics too, but they're not using living organisms or studying life to try to understand them. And I think that's kind of a misstep, because life interacts with, has so much interaction with information, with entropy. So I think that there's a deeper mystery to be solved. It's funny because everybody thinks that they have the something to say about it. And when you look at the sum total, it seems like there's a picture, but it's still very ambiguous and it's not really clear what's going on.
Do you feel like contributions from all levels, though, are part of the whole story? Like if you want to say where did life come from, you have to talk to...
I think so, and I think it's important to do that because if you look at there have been tremendous insights from say cosmology about the origin of life. In particular, a lot of these molecules that we'll talk about and however they emerge, some of them have been known to actually fall out of space. And you can tell because they're space isotopes.
That is cool.
Yeah. So it's interesting. There's a lot of different things that could happen. And then
when you say fall out of space, you mean like how we're stars, we're all forged in the center of a sun, right? Like everything that's not helium and hydrogen.
A sun is going to be too hot for those complex organic molecules to form. They would likely emerge.
Organic molecule. Yeah, sorry, I was thinking of the things on the periodic table, but you're talking about stuff that's organic.
Yeah, like organic molecules.
Okay.
Everything on the periodic table comes from stars or exploding stars. Anything else is gonna, those complex molecules, they need to be in a much cooler environment. So that tends to happen in some kind of a solar system or a gaseous space that isn't quite hot enough to be a star. And so I am the more I learn about this, the more I'm tending to think that organic biological life will look very similar in lots of places. I didn't used to have that view, because it seems that there's lots of different kinds of biological molecules you can make. I mean, carbon could do so many interesting things.
Right.
But when you look at the kinds of molecules that occur naturally and the kinds of things that seem to emerge naturally, they're very consistent with what we're made out of. So I tend to think that if life does evolve, independently, and if for example, there's life in multiple galaxies, it had to evolve independently, because there's no known phenomena that we know of that would, connect our galaxy to another galaxy, right? Because
we're asteroids or something, right? Don't they just
too
fly
far
away?
Even that.
Do you think that the way that life evolved here would happen the same way, just in a different setting almost, but the same process?
Wherever it happened, it could have happened multiple times and it would be apparently similar, but I believe. But it's
how are you talking about similar, in the sense that on the cellular level, or how
DNA, it would have RNA, and it would have proteins. Okay. So I believe that any organic biological life would exhibit would have.
Do you think
those
everything would
things.
look like a tube? How everything alive right now kind of in the mouth, out the butt.
Actually,
but do you know what I mean?
The digestive tract.
But like a lion, a tiger, a monkey, whatever you're talking about, it all kind of goes in and nutrients are absorbed and then comes out. Everything's a tube.
Yeah,
right?
it's efficient body plan for organisms that need to consume another, a dead organism or a still living one to survive, right?
Yeah.
If you're large enough, you've got all these cells you need to take care of. You have to go through a lot of trouble making sure that you have energy available for all of those cells. So, being a tube is kind of an efficient delivery system in its own.
So your guess is life everywhere in the universe would probably be tube-like.
You wouldn't see that all life exists as tubes because, not all life on Earth exists as tubes.
Yep.
Like, I don't think of most plants as being tube-like.
Nope.
Then,
Oh, yeah.
Sponges,
there's a lot of plants, too.
Things like that. There's
everything that moves even everything that moves isn't a tube, is it?
Yeah, so like...
Lots of tubes, but not everything.
Yeah, unicellular organisms, not very tube-like.
Or fungi.
Yeah.
Okay, we have enough examples, Matt, to prove me wrong.
But I do believe that the tube would come up somewhere else.
So what about the theory where life started at the bottom of the ocean from the heat?
The chemosynthesis.
Yeah, do you think that one's more likely? So maybe it wasn't actually the sun's energy?
So definitely. I tend to really like the hydrothermal vent theory.
What's that? Yeah, can you look that up? There's something on YouTube. Can you look at a hydrothermal vent on YouTube just so we can see what they look like?
Yeah, I like this one because you see this. It's an interesting example where you get like very volatile chemicals coming up out of the Earth's soil, and then it mixes with water. The other theory is like condensation pools. So if you would get concentrated substances in like a condensation pool, these hydrothermal vents are popular because you have these volatile organic compounds that come out of like thermal vents, and then it can react with water.
So a lot of inorganic material is flying out of these. There's a lot of heat, a lot of energy. So this is a theory that means that maybe this is where the right soup is stirring around at high enough temperatures to potentially create one of those like 1 in a trillion connections and make the thing that needs to replicate life.
Yeah. Additionally, there's a lot of energy there too. The hot water, the cold water, and then the
gradient between the two.
The chemical gradient.
That's a tiny volcano, right? Like this is hot magma and lava underneath the seafloor heating up.
It's all the gas from the center of the Earth coming out, all that byproduct from the lava and stuff.
See what comes out of a hydrothermal vent.
Particle-laden fluids. A venting black smoke emits jets.
Can you look up the anatomy of a thermal vent or how thermal vents are formed?
And then look up hydrothermal memes.
Buddy.
There's got to be a lot out there.
There's gonna be none.
Oh, bro, of course. Everybody jokes about hydrothermal vents.
So hydrothermal vents are a result of seawater percolating down through fissures in the ocean crust in the vicinity of spreading centers of the subduction zones.
Dude, I love a bunch of those words. Percolating, fissures. Do you like percolating?
I used to have a percolator that would make my coffee every morning.
Nice.
Oh, that's so cool.
Yeah, it's a great word, man.
And then I left it on and it melted. The handle melted off.
Oh my God.
Really?
Yeah, because I used to have a fissure that made my coffee. It was the best, man.
Rich people.
Thanks. Appreciate you laughing at that one. Okay, so we're going to look at this experiment. It's called the Miller-Urey experiment. A lot of people reference this when it comes to life. What is happening? Why is this experiment important? Okay. What does it tell us?
So this is a great experiment because it's the first one that demonstrated that there is a naturalistic process for biological molecules to come into existence. In particular, this was an experiment that showed that you could use ordinary inert gases to make amino acids and then later protein chains or amino acid chains that could fold into proteins. So you could see like the big glass chamber on top. It has the two metal rods going in, and then there's actually an electrical arc that connects them. That is the energy source needed to convert the gas in the chamber. So in this experiment, he's using N2 and CO2, whereas the original Miller-Urey experiment, I believe, used ammonia and methane. Now, it's cool that this experiment still works because the N2 and the CO2 are more consistent with what the early Earth's atmosphere would have been like. However, it doesn't really matter in my opinion because what it shows conclusively is that there's a plausible path for these organic compounds to come from, and this was the first person to show it. So, Stanley Miller and his.
So we start
boss.
with all inorganic material, stuff that you would not consider alive, and you end up with the building blocks of life.
And the only ingredients he needs are there's water, there's nitrogen gas, and there's CO2.
Transfer this into the test tube.
It's gonna explode. Oh no.
We're going to add 12 drops of anhydride.
What is that, CBD?
Oh, this is awesome. So that is the building blocks. The proteins are in there?
Well, so he's adding a reactant to indicate that they're there.
Yeah. Okay. So it's there. Well, because we know it's going to react.
So what he does next is, I believe, he has to heat it.
But the heating, when it actually formed the amino acid, they should be in there already, you think?
The heating has to do with
proving that they're in there.
Yeah.
That's just making the reactant dye them correctly.
It's looking pretty good so far.
Oh my God, the liquid's turning black.
And what does that mean, Matt?
I don't know, man.
It means life finds a way.
Oh, okay.
We just created the building blocks of life from inorganic material.
That's crazy.
And there you can see the nice dark purple color. That is indicative of the presence of amino acids.
You debunked the debunkers.
Debunkception.
Isn't life 4.2 billion years old or something?
So the Earth is 4.5 billion years old.
Life shows up relatively quickly, right? Like only a billion years after it formed or something? Fossil record, or like when did life start? 3/7. Okay, gotcha. Okay.
Almost as soon as the Earth was cool enough.
Yeah, surprisingly. So it almost makes you, but the reason I think that's interesting is because it makes you wonder, like as soon as everything shows up and it's right, does it happen?
Well,
like,
let's think about significant figures here, because we're quoting a number like 4.5 billion years, and 0.5 billion years is 500 million years. So like, 1 million years is, doesn't show up in that figure. And that's a lot of time for life to evolve, for things to happen.
Like we have amino acids, and how is that correlated with living, breathing mammals and dinosaurs and fish?
Amino acids are they form the monomer units that you can chain together to make a polymer peptide, which, when you've got a polymer peptide that's even longer, if it's dissolved in water, it folds into a three-dimensional shape. And we call these proteins. They're important because of their catalytic and mechanical abilities.
Dude, Microbial Minute, let's sign up.
Today we'll be highlighting a paper from Science that provides a recipe to synthesize some of the basic building blocks of life. Now, one hypothesis for abiogenesis, or the formation of biological life from non-biological precursor molecules, is the RNA World hypothesis. This posits that RNA molecules were the first biological molecules that formed in that primordial soup. The wet-dry cycling around these shallow ponds may have provided the necessary conditions for specific chemical reactions. Previous chemical studies from this same lab, in fact, have generated the purines, the adenine and guanine, from early Earth conditions.
By evaporating, the evaporation pools would make a denser pool of,
Just brings it closer together. Okay, gotcha.
Reactions also assume a fluctuation between both acidic and basic conditions, which the author suggests could have come from acidic rain, acid rain, and carbonates, respectively.
I see.
The wet-dry cycling may have occurred due to the seasonality that was present even in early-day Earth. This model assumes that these reactions did take place on Earth's surface, which is in contrast to other models that have posited an aqueous beginning to life.
So she mentioned the 4 different nucleobases that you could get. They come in 2 different families. You have the pyrimidines and the purines.
Nucleobases means what? Like a group of... So we don't have nucleotides yet. As far as I can tell, we only have protein, or we only have amino acids.
So we have we had the amino acids from the Miller-Urey experiment. What we made here were the nucleobases. Now, DNA and RNA have 3 components for each molecule in the chain. You have the phosphate group, you have the ribose sugar, and then you have the nucleobase. The ribose sugar and the phosphate group form the backbone, so-called, whereas the nucleobase forms the pair bonding piece that complements another strand, or for that matter, in the case of RNA, it's the one that does the
right. And our Urey-Miller planet doesn't have that yet on it.
No, it wouldn't be there. So,
but it would have the phosphate and what makes the backbone? Out of those 3 things, 2 things already exist.
Phosphate,
right?
and the ribose form the backbone.
Does ribose exist yet?
Ribose is a sugar, and that's they talk about this at the end of the
sugar. Does sugar exist without life?
They talk about this at the end of this video. Is that's their next? This researcher's next step is to try and find the origin of ribose.
So we have to figure out where sugar came from.
So Yeah, so this is what she was talking about. She called it the Krebs cycle. But this is a citric acid cycle, and it's really important for making ATP and, well, basically breaking down sugar into more manageable molecules that your body can use. Because sugar would, if you tried to burn sugar as an energy source, it's actually way too strong.
It's all locked up.
Yeah, and it would cause a lot of problems for cells. So instead, you have a very careful process that's used to break it down. Now, there's some researchers, Dr. Schiller, you've met, he's argued this one to me, is that the citric acid cycle is such a fundamental, it occurs using such fundamentally small pieces that, and it's just so, I don't really know the entire chemistry argument behind it, but there are some chemists who believe that the citric acid cycle in particular is a common feature of any organic life. It would have to emerge independently for any such organism. So fun stuff.
So,
but going back to go ahead.
To get this right, this is basically the key to unlock energy to actually be used.
This is definitely a important part to the origin of life.
Okay.
This is an interesting experiment. A guy was looking at the role that boron might play in the silica of the glass in catalyzing these reactions. That wasn't originally considered, and up until this guy came along, it wasn't considered. Yeah. It should be terribly surprising because the glass that you see in this, if you look at like the clays that life would have been, or these early molecules would have been exposed to, you look at clay under a microscope, it actually has a very crystalline shape. And as such, it's, clay being silica is also going to have other mineral impurities, boron plausibly one of them. In the earlier video we were watching, she talked about zinc. But there's many kinds of, metallic catalysts that could be, in, soil or such.
Yeah, so that's why maybe on the surface. So the surface of Earth is starting to sound a little bit more ripe than I was thinking.
But there's also soil and hydrothermal vents. Look at this. Look at all of those all of those molecules that he was able to pull out of this. These are all organic molecules that he was able to verify or that were produced by his replication of the Miller-Urey experiment. Many amino acids, and get this, several pieces of the Krebs cycle, or the citric acid cycle, also known.
Coming, dude, that's a huge key then, right?
So scroll down, let me see that list. So looking at the amino acids, we see glycine, alanine, valine, leucine, proline, serine, asparagine.
Matt, you know what this means?
Aspartic acid,
it means so much
glutamic acid,
things.
lysine, histidine. We also have adenine, guanine, uracil, cytosine. These are
they're things you hear all the time.
These are
in regards to our life.
Submolecules of DNA and RNAs, right? And then also there's a bunch in here that are related to the Krebs cycle. You have lactic acid, fumaric acid, malic acid, oxaloacetic acid, alpha ketogluteric acid, and maybe another one in there somewhere. Succinic acid down at the bottom, pyrene. Yeah, let's go back to the citric acid cycle.
So this was a big aha moment for you when you
and
read
so,
that.
Yeah, and there's see these alpha ketogluteric acid, succinyl acid, succinate, fumarate, malate.
Very good.
This is really cool. I
Oxaloacetate.
see why this is important. I mean, that's really a lot of things coming from a place that can be tracked all the way back to inorganic material, and things that are very uncommon.
But it's not just as simple as the 20 things that the original Urey-Miller experiment kind of produced. It's so much more.
You know what?
So it helps.
The original Miller-Urey experiment, as it was reported only like 7 amino acids were synthesized. It turns out that was just a feature of their low effort in detecting all the different amino acids that were probably there. They had followed up actually using leftover bottles after Stanley Miller died. They reanalyzed them and found 17 amino acids and then chains of amino acids. So it wasn't just the 7.
There was
a lot more
that came out of that.
Yeah.
Wow.
I mean, that was wasn't that like 50 years ago or something?
Or more, yeah.
So how come like every grad student's not doing Urey-Miller? Like, how come we haven't built hundreds and hundreds and hundreds of replications of this? Or have we? Like, I'm just surprised like not everybody's like knows this and redone this.
So that did happen, and what happened was, like, I think after the Miller-Urey experiment came out, some scientists thought, hey, what can we do with these, these molecules once we have them? Maybe if we put it in an oven and bake it for a bit, it'll do something interesting.
Just playing with all the variables of what's in there and what can happen next.
And it's been a slow process in making like these small, molecules. And it's, because, life is made of very big molecules. We're made of, proteins that could be anywhere from, just a few amino acids long to tens of thousands. I think there's one that has like 10,000 residues in it called Titan. So it's
huge, yeah.
It's a muscle protein in people.
Right, and this is where when you start thinking about the probabilities of that all coming together, you need to think about something that could maybe kind of help guide that process. That's it. That thing, like, to me it's just like a cool colored thing, but to you is that just like a gigantic?
Like,
are you like, that's like the Giganotosaurus of DNA?
It's a bit of trivia. I know it's the biggest protein I know of.
That's a lot.
Yeah, pretty big.
You guys know the longest word in English?
No, what is it?
It's pneumono ultramicroscopic silical volcanoconiosis.
Oh, good for you, man. You know
That's
that?
a diamond disease.
Oh.
It's a what?
It's a disease you can get if you work in a diamond mine.
No.
The diamond mine, then I don't know. Hey,
Okay.
You can check that out for us, Matt, while he's talking.
Oh, how do you spell it?
Oh.
You know what the word is, but you can't spell it? Come on, man.
Like PNEU something. Pneumono ultramicroscopic silical volcanoconiosis.
A lung disease.
Imagine you have a bunch of Legos on the ground, and I want to form a boat, but I want the boat to actually float in the water to go somewhere. Like, how did we go from like a bunch of stuff into things that want things, that interact with each other, that don't want to get dead and want to get energy and move around?
So it sounds like you're really asking about like emergence, and how do we go from these simple molecules to like these the emergent properties of replicators of
because like with
meaningful things.
Yeah, just to put it in my brain, the Legos, you could stack them forever and they could become boxes and random looking things. But like, when do they become a boat? Like, when do they become like an airplane, a city? Like, when do they become a thing that exists in the world, that wants something, that does something, that has it's
purpose.
Guided in some way, some kind of purpose.
So, a lot of those concepts that you brought up, like boats and cities, those are not easily explained with, the origin of life.
Yeah.
No. So, okay, there's some interesting stuff about like self-assembly and these emergent properties, is that self-assembly can occur if you settle on a few simple rules and just let them play out. And there's lots of different simple things that you can let play out in this way. Conway's Game of Life is one of my favorites. It's very, very famous. You basically start with a cell, and there's a simple rule. Like, the cell is either going to live or die. It lives or dies based on how many cells are around it. If I recall correctly, it's if there's no cells around it, it dies because it's starving. If it's surrounded by cells, like 5 or 6, then it'll die.
Right.
Well, because it's suffocating.
Yeah, I forgot the pattern too. But
if it's
we're essentially thinking about a grid, like a checkers board.
Yeah.
And
if it's 2, it'll increase to 3, and then it's like that. But anyway.
But even if we don't get those specific rules, the concept is that, imagine a checkerboard, and you might have like a red checker, and then depending on what it's surrounded by, and it can be something very simple, you can end up with extremely elegant emergent patterns.
Yeah.
If you just follow that same simple rule, whatever it is. And let's say, I don't think this is it, but let's say if the checker has an empty square to the left and a filled square to the right, and it could be just that simple, and then multiply it. And these are some examples of the kind of designs you get from incredibly simple rules.
Yeah. So some of these have names, like the glider. The little tiny ones that it's like, I think it's only like 5 pieces, and it kind of dances and kind of moves across the screen. It glides. It looks like a glider.
Yeah.
And yeah, it only takes these few simple rules, and then from this, you get these kind of almost lifelike patterns that emerge. What's really cool about
look at that one.
what I think is really cool about Conway's Game of Life is it's complex enough of a system that you can use it to program a computer, to emulate a computer. So it's Turing complete.
What?
Yeah. That is, you use a computer.
I did not know that. It's
to emulate
Turing complete.
Conway's Game of Life. But you're going to use such a big Conway's Game of Life that it can actually emulate its own. It's going to be a computer itself. It's doing computations.
Yeah. Okay, I guess so.
That's
I mean, because
insane.
computers are made of really simple logic gates. So if you just hand something like an AND, an OR, an XOR, you can program something complicated. So I guess simple rules. Yeah, why not? That's crazy. That's crazy. I hadn't really thought about logic gates and computers in terms of Conway's Game of Life. That is really cool.
This is Conway's Game of Life still?
Yeah.
You should. So when we're talking about simple, when you think about simple rules emerging, can you throw information into that? Like, how do you see information? Where does information start to play a role? Because it doesn't feel like the Miller-Urey experiment is information. Like, there's not patterns there that are anything. Like, we're not talking about RNA or DNA that hold any serious information generation to generation.
So the Miller-Urey experiment there is information in that because you have molecules that are more complicated than the ones before, and it's significant. I mean, you saw the experiment where the guy adds, he has the indicator and then he sits it in the hot water and then it turns purple.
Right.
That was information. The change in color of purple indicated that there were different molecules in it than there were before. So there's this information.
But what about the iron and stuff that was sitting on Earth before life?
So,
you could have catalyzed that with something, and it wouldn't be information, would it?
Well, I mean, why wouldn't? So information is
because it would have been pure randomness. It would have been because some heavier elements are less common than others. Is that information?
I mean, yeah, you just gave me information about elements.
But the periodic table.
You just
Wait, so information did exist. Okay.
So
information obviously has existed long before life. Okay, dumb question.
So information is a complicated subject. There's apparently a relationship between entropy and information, such that entropy is information. That you can get like these Miller-Urey, like going back to the Miller-Urey experiment, you get these amino acids, and if you run the Miller-Urey experiment, these amino acids will sometimes actually chain together. There's information in that. That chain reveals the order of operations of how that molecule must have come into existence. If not the explicit order of operations, it tells you
something that is more than random.
Yes,
it's even if more specific than random.
Even if it is random, though, even if it's random in that it could have been many other ways.
Yeah, there
is the way that it is, and that is the information.
Okay, and just to put this in context, so how familiar are you with entropy? Because it's a measure of disorder. So in general, a deck of cards that's in perfect order, if you shuffle it, it's going to get randomized over time. So when you have the Miller-Urey experiment, you're actually taking what is all these random molecules and they're starting to chain up, which is essentially like shuffling a deck of cards in such a specific way that it's starting to line up. So you're now fighting entropy. You're now getting more organized. And humans, life, everything here is we're surprisingly going the opposite direction. We're taking energy and organizing it into our DNA and our.
But there's a paradox there in that energy or that entropy actually comes, or that information comes from entropy. Like it's bizarre.
It's
information
the information that can't happen almost, or the options that don't result in
Well, the information goes away when you shake something up and all the order is lost.
Yeah.
Mm-hmm.
Like a box with like a Lego boat in it could be organized to do something, and if you shake it up and all the Legos fall apart, so the information has been lost. It's moving towards disorder.
So
right?
I disagree. I don't think that's how it works.
Okay.
There's in a random thing, something that's random, you would actually say there's more information in it because you have to record more to reproduce it. So if I give you a very orderly, stack of Legos, for example, for you to reproduce it, it's very easy, right? But if I give you a pile of Legos,
right,
randomly thrown across the
because
floor,
if it's a random stack of Legos takes a ton of time to describe. Whereas if it was like every third one was blue, you could have a simple rule.
Exactly.
So that actually has,
and it's paradoxical.
that has
because-
less information in it, even though because it's got a pattern.
Yeah, and it's paradoxical because the random arrangement is, one of many random arrangements, and we think of that random arrangement as all being like, okay, well, I'm going to dump these Legos on the floor. It's going to be just like, Dylan's mess of Legos on the floor.
Yeah.
Yep.
And but it isn't. It's different.
And it's hard to
Fascinating.
describe something because it's so unorderly.
There's another one that I love. This one is actually a little more physical. There are these things that they call Janus particles. So basically, Janus, if you've ever heard of
No, I've never heard of it.
Janus is a god with two heads, like a Greek god with two heads.
Oh.
And basically, with these Janus particles, what you're going to do is you're going to have like there's usually a positive charge on one side and a negative charge on the other. Alternatively, what you could do is you could have like one side that I guess is aluminum, so it doesn't react with anything, but the other side is going to be platinum. So it'll actually catalyze certain reactions, and then they'll take these little platinum beads or these half platinum beads and drop them in like a hydrogen peroxide solution. The platinum will catalyze hydrogen
Do
peroxide
they form interesting shapes?
into water and oxygen, and that the oxygen beads that form off or the oxygen that now as a gas starts to form on it will push against the bead, and it kind of drives it around. The other approach is to use
more like Conway.
charge.
So it's more like Conway's Game of Life. You'll see motion, or like maybe those like the little airplane or something will emerge.
Crazy.
Not quite that cool, but actually let's.
And so the aluminum is like the direction that it's going. That's like
The aluminum would essentially point in the direction that's going to go.
So when we were talking about a few rules in the Game of Life, because these things kind of have a polar side and an inert side, they kind of make rules, and when you jostle them together, they become
They do something
more
orderly,
than
because
the sum
they're-
of their parts. Kind of they.
Yeah, well, since they're all following the same rules, they may form something orderly together.
Yeah. Oh, cool. So So complex can emerge from simple.
Yeah.
In mass.
So there was let's see the video that I found of the flagellar dynamics. This one doesn't use this one uses the charged flagella, the charged Janus particles.
Oh yeah, you can kind of even see the Janus particles, little yin yang guys running around.
Yeah,
Whoa.
and what they're doing is they're using a magnetic field to induce these.
Flagella. This is what's in our cells that move. Oh, because I forgot from biology. That's what our cells have, the little flagella. That's how they swim around.
Mm-hmm.
Oh, and they can come, and those things can come from something that simple.
So
now we have a motion. So now we have an inorganic material that turned organic, that clustered into something, and now it can move.
So these, I mean, yeah. It
We
moves
made
based
life, right?
No, not quite.
Come on, we're gotta be close. That thing looks like it moves around.
I mean, it moves around and it's cool, but we're just talking about like,
okay.
self-organizing and emergent behaviors.
It looks like a little snake, man. Beware. Here comes your Janus snake.
But there are similarities. Look at that.
That's the cool stuff.
Oh.
And they're all following like a magnetic field.
Will they eventually just
Oh.
become one long snake, or do they have a tendency to not?
They have different patterns.
Well, yeah,
Good
but
question.
they're
Not
all
really
made of the
sure.
same thing. Won't they eventually just all connect?
Well, they look like they're kind of. I mean, some of them connect and then they
Oh,
disconnect.
and sometimes,
then,
So given enough,
they'll even like hand somebody off and be like, "Hey,
Yeah.
why don't you
Okay.
take this guy now?"
yeah, okay. So then you get a lot of different. I mean, it looks like a little bit of Conway's Game of Life.
Yeah,
it looks
so,
like something complicated could emerge from, given enough time, for sure.
Yeah, so physicists love to play with these. Yeah, they do some really good visuals here.
Oh, it's a pattern right there. Yeah.
So that video is just explaining how the cells interact with each other based on properties of what they are.
So it's not necessarily about the cells. They kind of look like cells just because they're
We're
round,
not
but
too-
Not
cells, yeah.
not cells. I mean like the molecules, how
They're
they
called-
interact with each other.
What are they called? Janus molecules?
Yeah, they're Janus particles, and so it's an analogy for how the simple rules, whatever, even very simple rules, you got a positive charge or a negative charge. As long as all of these little entities are following the same rules, they may do something very complicated.
Mm-hmm.
Okay.
Yeah, especially
so it's just the advancement
if they're
of
following the
simple
same rules, maybe working together, so to speak. Something more complicated can emerge.
Yeah. I love
Okay. I'm connecting the dots.
Let's look at the self-replicating ligase ribozyme. Okay, cool. So actually, this is just a link to an actual picture. So we talked about this in one of the reaction videos a little bit, right?
Yeah, but they might not have watched that in the podcast.
Cool. Okay, so
assume they haven't.
Okay, so this is an example of a RNA ligase replicating enzyme, or ribozyme as it's called. And basically what it is, this is a two-dimensional description of the folded structure. You have, like, if you zoom in right there, see the PPGOH? Okay, so that OH belongs to one of the ribose groups, and it is part of the mechanism that this ribozyme will use to catalyze other residues to replicate itself.
What's residue mean? I don't understand.
So residue would be like each element in the chain. So each nucleotide in the chain. So that's the active side of the enzyme, basically.
So that's the area where the replication would happen.
Yeah, and by the way,
could I imagine like Janus particles coming together eventually and creating something like that could then catalyze two things to stick together? Like they eventually could figure out how to glue two things together.
I don't know how you would get Janus particles to do all of this, because this is RNA, and
okay,
RNAs,
so now we've missed a step then, right?
RNAs, yeah. So there is kind of a leap there, like we talked a little bit about like the RNA molecules and how are we able to get those yet without any too much help, and we can get the nucleotides. We're still like trying to get the whole thing. This is a whole 80 residues all chained together in a particular sequence. So there is a lot to get. There's a lot to bootstrap up to. So, and these kind of things are isolated from living organisms. Usually.
This is where we need to pour billions of dollars of research.
Is that what you think?
I mean,
Yeah,
right?
maybe.
This is the gap to figure out.
Yeah, I mean, and like the RNA is really, really crazy. We used it for the most recent vaccine for like COVID. The Moderna mRNA vaccines. Yeah.
Mm-hmm.
There's lots of interesting possibilities and things we can do with RNAs. This is how it works. Okay, so you have the molecule, the whole chain. It has to dissociate from itself to replicate itself. And you see a process here where it breaks off into like an A and B chain, and then the A and B chain can recombine. It synthesizes its complement, and then you see like the opposite complement that can go off and separate to do its own replication, and thereby create the original.
So it separates, connects, separates again.
Yeah.
But it connects with another one of the original. So there's two that are broken off, but it connects with another one, or is it the same one in the beginning that reconnects itself?
So
it's a mirror, is that right? It's a complement.
You have to
it's a left hand, right hand, then back to right hand?
Basically, yes. So you have to use your left hand to make the right hand, and then once you've got the right hand, you can now make another left hand.
Gotcha. Okay.
But in this case, the A and B pieces actually separate. I think that's actually the active site. If they don't have the complemented piece, then it doesn't form the ribozyme. It looks like.
So to wrap my pea brain around this, Janus particles.
No, you are very smart.
Those are to show off like how simple things can create complicated things. That's like the main thing for Janus particles, correct?
Yeah.
It doesn't necessarily
It's a main
hold weight with
concept.
amino acids creating DNA.
No, yeah, it's just like another way of how
To show it's
rules-
possible.
Yeah, how simple rules can emerge. You can get cool patterns.
Okay.
Because a Janus
that
particle
makes sense.
is something created in a lab. That's not ever going to be found out in nature. But it demonstrates how nature might have done something. Is that right?
Yeah, basically.
Janus particles are lab creations.
Yes. And that, but like I said before, if you have lots of entities that are all following the same rules, they may work together to create something, and something much bigger will emerge.
Yeah.
So.
Okay, yeah, that helped me understand this
Yeah,
way more, honestly.
I guess like it seems interesting that Janus particles are used because they're actually manufactured to be more simple than life. Life would probably have had something with a bit more variation and more tweaking, which actually would be more likely to build into something complicated. This is like the simplest gate possible, and all this can still happen. But
Yeah.
you might even have something that's a little more complicated, and then you could have even, nature would be more creative even. This is like as dumb as you can get, and it still works.
Yeah, Janus particles are the way to just illustrate the rule of
It's
simple to
like
complicated.
it's like the toy, the canonical or the toy example, just to prove it out.
Now,
Okay.
if we want to talk about bootstrapping up to that complex ribozyme,
For sure, I do.
Let's look at the Minimotif.
Do you think we're gonna let you out of here without the bootstrapping up to the more complex?
Let's talk about that Minimotif.
No way.
This was written by a professor I used to work for, Dr. Schiller, who you met before.
Yeah.
Minimotifs are short peptide sequences as part of proteins that are really important for protein-protein interactions. So they're basically just going to be 2 to 15 amino acids in length, and that's it. And they're really important for signaling about proteins. You can tag a protein for destruction. You can have trafficking signals attached to a protein that tells a cell where to take the protein. Okay. And then also you have some catalytic abilities, if you phosphorylate a protein, you can that's a Minimotif. So the phosphorylated residue is a point that you attach a charged phosphate to, and that charged phosphate will cause the protein to change shape, and you can get work done out of that protein by phosphorylating it and then dephosphorylating it, and it'll work. You can treat it like a pump. In fact, your body has cells. All your body cells have pumps that work essentially just like that. You phosphorylate it, you dephosphorylate it, pump runs.
And it moves things
So
around.
it connects, disconnects, and then reconnects.
Yeah, and because that charged group
And
causes it to change shape.
It's actually making
pushing-
sense.
But anyway, what we're looking at right here are these. Actually, imagining these Minimotifs as mediating these chemical reactions all on their own, not as large superproteins. So what he's kind of conceived of is that you could have little triplets of amino acids, and you only need 3. Really, a chain of amino acids, just 3 long, can fold into something that can actually touch itself, end to end. It's not. They're very rotatable, and if you could get a couple of them to work together, maybe opposite of each other, or in the case of one of these, there's a high affinity motif that's used to bind to something. It just kind of folds in on a target. This would be a theoretical way to get started up to that point. So I would tend to think that the Minimotifs probably come first before the RNAs, and the RNAs come later because they're so much more complicated of molecules. Because these Minimotifs, it's just the amino acids. Each of these amino acids, they're actually smaller than the nucleotides, the nucleobases themselves. The nucleobase itself still has to have a phosphate group and a sugar group attached to it. So, RNAs and DNAs are much bigger molecules. These are cool little intermediates that could, plausibly help us get up to more complicated.
So what experiments or what evidence, or do you see anything like a Minimotif in the wild? Has anything been done in a lab that gives you some understanding of how it could
Dr.
bootstrap?
Schiller works extensively with using Minimotifs in biomedical research. I would have to let him talk to you about that. And as far as, prospects for, using this to, help enhance the Miller-Urey.
But as of this point, there's not a Schiller experiment that's
haven't seen it, no.
this could be this gives us the probability, gives us something to.
I haven't seen it yet.
Okay. All right.
This is so cool.
So it's our best.
Wow.
So this is where, so see, Nick, you got to devote your life to this.
Yeah, you should.
Imagine you figure out how these things bootstrap life. You go down in the history books.
This is so cool.
Maybe they're the key. Is there a competing theory? Anything else besides these Minimotifs that you think might be in between the RNA and the amino acids?
So the RNA world is the most famous theory. It's most widely held because RNA is self-catalytic, and it is also a great
Wait, but RNA. Wait, explain the RNA world hypothesis, because it sounds like we don't have RNA yet. We only have amino acids.
So the RNA world hypothesis is important.
But it's too high level.
It seems like it might be a little complicated because the amino acids appear to generate very easily, but we still don't have great experiments that show these chains of nucleotides emerging naturally. So I mean, that is a fair criticism of the theory, I think, but it's still widely held because if you look at like the three molecules of life that are really important, you have your DNA, you have your RNA, and you have your proteins. DNA is great for storing information, but it can't catalyze anything. It's very poor at catalyzing any other interactions.
Its asset is robustness.
Yes, there you go. Proteins, on the other hand, they tend to fold into these elaborate shapes, and then they're highly catalytic. They're very active. They cut each other up, and they do all sorts of fun things. It's a poor energy storage molecule because if nothing else, it folds a lot, and that particular helical structure of DNA helps make it a very great information storage molecule because you can unwind it, and it's the whole thing's a very regular shape, whereas proteins don't have a very regular shape.
Yeah, nothing stiff there to keep them together.
So real quick, going back to the minimotifs. So those are all just amino acids, and then they just start kind of coming together, and so then more and more and more, and they just kind of bundle up all together, and that's what creates a protein.
So a protein is going to be a chain of amino acids.
Okay.
The minimotifs are the shortest protein chains you could conceivably have, but to make a bigger protein you just need more minimotifs, or just more residues.
And so what makes them like want to come together? It's just how they are. They're just attracted to each other. They just know that they fit, so they go that way.
Okay, so amino acids interact with each other largely through charge. So the hydrophobic residues, they avoid water. For the large protein structures, that tends to be a big part of how the protein gets its structure. The hydrophobic residues will orient inwards, and then the hydrophilic residues that like water, that have the charged sides, those will orient outwards. Now, as far as how proteins interact with each other, like I kind of mentioned the minimotifs, the minimotifs are often targets of protein interactions. So like, if you phosphorylate a minimotif, as far as other things proteins do, they're all different.
Yeah, that was such a good question you asked, because I never really thought about how do they bundle up.
Different things. We have to settle the issue of like how to get amino acid chains. And then there's also the things amino acids do. The amino acid chains are, they're formed from there's the amine group and the acidic carboxylic acid. And they just bind together and leave a third, so-called R group out that is allowed to interact with other things. That R group is going to be the charged group or the not charged group. And you're looking at some of these amino acids. They have a couple like a negative charge. So if it has a negative charge, it means that there's a couple of electrons there that are active and have something to do, or
that attract other.
They're
amino
looking for
acids.
something to do, and they're gonna attack another molecule or something like that. And I mean that's an actual term they use in organic chemistry. Attack.
That's so cool. It all makes sense.
If we have these protocells, which I'm going to just define as like not quite cells yet, but they might have the elements of a cell and they could come together, how might they be replicating?
So I don't know where I heard this idea. I'm sure it wasn't mine. But I kind of like the idea that like if you're going to use the hydrothermal vent theory, you could have these organic molecules that would, I mean, if you could get the RNA protein or the RNAs, that backbone or whatever, or sorry, that instruction set. And then you also have like some proteins with it. You could get particles that would, maybe like almost like a viral particle that could be stable enough to transport to another hydrothermal vent. And that would be the first step of something lifelike that I would consider. Because like if you had like these a mostly inert, cluster of molecules that, as it travels between like, hydrothermal vents, it does nothing. But once it gets to a hydrothermal vent, it's now hot enough. It's now has that environment to interact. It'll start, replicating itself. It'll start to interact.
It's a mixture of resources.
That would be the first pseudocellular thing I could think of.
You know what's crazy about that is that when you described it, it sounds like the way I've heard people describe a virus. Just kind of like floats around. It kind of can hold its own container and keep some information, but when it lands in a certain spot, it can do a thing. So now you're talking about something that maybe that's jumping from like hydrothermal vents or maybe jumping from puddle to puddle. But I don't know. It has that, like, life definitely seems to like that model because viruses are much more abundant than cells, than cells, right? Isn't there like an incredible amount of viruses just sitting around the planet?
I want to say they outnumber us 10 to 1 at least.
Yeah,
Oh,
like
just in cellularity. There's there's more viruses than there are cells.
How many more viruses are there than cells or something? But anyways,
And that's because they're fairly simple in the fact, and then
they're just keys sitting there waiting for a lock.
Whoa, look at that.
Oh, 10 to the 8 viruses to match every cell in our body.
Wow.
You're like 10 to 1, like 10 to the 8th to 1.
Wow.
So, I mean, that's what's
I
crazy.
would not-
So, like, you
I
already
didn't-
think, like, our body
oh my
has a trillion
God.
cells or whatever, but then whatever that is, for a trillion cells in our body, there must be a quazillion viruses floating around in this same environment. For the fact that we have 4 human beings in here, if we each have a trillion cells or something, how many cells are in the human
There's
body?
a trillion viruses.
Yeah, so there's a trillion viruses in this room right now?
Is it because cells are living and viruses are
Guess
not?
so.
Look at that. It's 37 trillion cells. That's not that much.
No,
Oh my God.
You have
That's
to do,
per cell, though.
So now
That's how many viruses there are for each cell in our body.
yeah,
And we have
And
a
there's
now that times
37
3.7 billion. I mean
3.7 trillion.
billion. Oh yeah, whatever.
I don't know how to do a trillion. It's a big number.
You
It's
put a
every-
dot instead of a comma.
1, 2, 3, 4, 5, 6, 7, 8, 9. That's billion, right? Trillion?
Yep.
Yeah, I think so.
Well, close enough. So 3 in the power of 20.
Okay.
My brain can't even compute
I know, but
how
it's
much
just, it's
that
just
genuinely
insane to
is.
think.
massive.
Yeah,
It's
like
massive.
and
It's a massive number.
where are they? On the carpet, in the ceiling, in the
Everywhere,
air,
man. Everywhere.
Yeah.
Everywhere.
and they're just waiting to get into one of my cells and infect me. Do you think a virus is alive?
I actually think of viruses as living organisms.
So
not
that's
as
a living-
not as viral particles because they're not doing anything. But if you look at it like a host cell that's been infected with a virus, in what respect is that host cell not now belong to the virus? In what respect is it not a new organism? Because like, have you ever heard of the HeLa cells?
No.
You haven't heard of Henrietta Lacks? Okay, so
Oh boy.
Henrietta.
Is this basic
No.
stuff I should know?
That was somehow Google caught you there.
Like how to?
Google just loves Matt, I guess.
So there was this lady from back in the I think 1950s, 40s, 50s. She was attending. She was
Yep, 1950s.
yeah okay yeah. So she was a patient at Johns Hopkins with I think it was ovarian cancer, and what they did it wasn't ovarian cancer. It was uterine cancer, and basically the researcher biopsied her and
Cervical cancer.
to prove that she had cancer, and then he did something kind of nefarious. He took the cells without her permission and tried growing them inside a test tube, and it worked.
Whoa.
And then he shared those cells with every scientist on Earth. He started producing them like in the lab and like growing them in the lab, and then using
And
them experimentally
giving them
because there's never been like nobody had ever been able to grow
It's like-
human cells in a lab before. But these cancer cells, you can grow in a lab because cancer cells are just so
resistant.
they're just I mean they're broken cells. Like most of your body cells, just to avoid being cancer, like they have a default setting to die if something goes wrong.
Apoptosis.
Anything goes wrong, they kill themselves, and that's their job.
Oh, right. Is apoptosis a default setting?
Yes.
I assumed it was like a signal, but you're
No,
saying
it's a
like if
default
things aren't
setting.
right, we just die.
If you cut a piece
Oh,
of
I didn't
tissue
know.
out of you, I mean, arguably it dies not because because all the cells are programmed to die. So I mean, it's
replicating correctly.
yeah, having a default state of self-suicide is a great way to avoid getting cancer.
Okay, so this guy found some cells that were breaking the rules and sent them everywhere.
And we call them HeLa cells. They're the most famous cell line on Earth, I guess. It's widely used in biomedicine all across the world today.
And cancer research.
There's no going backwards from this. But yeah, that taken without a permission, really sad.
Yeah.
Also a groundbreaking development.
That's why.
Sure.
So what do you think about, like, do you have an opinion on gene drives and if we should like release them out into the wild?
Gene what?
Drives, like where you take like a mosquito and you modify with CRISPR its DNA so that
that's fun.
It can't have malaria. Then you put it out into the wild and it mates, and that passes into all of the next generation, and all of a sudden we cure malaria.
So I'm not terribly worried about.
Gosh, we should do a whole
trying
other
that-
episode on CRISPR though.
Yeah.
I know quite a bit about. I've been like reading a lot about Jennifer Doudna's work.
You'll have to find somebody who knows about CRISPR, not me.
You're not the CRISPR guy? No. You're my biology guy, man.
Not the
Closest I
CRISPR
got.
guy.
Okay, anyways. What's your thoughts on gene drives?
I think it's a great and interesting approach.
These mosquitoes exist, by the way. They're in a lab right now. Hopefully they don't escape unless you want the world to not have malaria, which I do. So I think I hope someone kicks the wall down and releases those things. But
the thing is with that, it just gives out a lot of like ramifications in the end of what could happen if it's used nefariously.
Actually, yeah, I don't want someone to release it without supervision. I want it very, very carefully studied and very carefully let out. But I think the risks just outweigh the rewards. And like life would do it. I mean, like without us, if it just life does all sorts of weird stuff and it just goes all over the planet. I think we should release these CRISPR-Cas9 super.
I actually I thought of this many years ago because I was reading a book about genetic drive. And I was like, how cool would it be if somebody put this in a mosquito or some shit and then released it out into the wild and get rid of a bunch of mosquitoes because all they do is cause malaria. I tend to be for it.
Yeah. So so I guess like the quick summary is the gene drive makes it so that it sort of purposely makes it into both the next generations, and you can't get rid of it. It'll propagate into the whole population. And the way mosquitoes breed, you might get like 99% of mosquitoes covered in a reasonable amount of time. And there's also a mouse, I think, or a rat that has it. There's another gene drive with a rat that can't.
haplotype.
You do something. I don't know. What is that?
Basically what happens is when you have one of these haplotypes, it's a huge like segment of your chromosome, and it prevents like you won't be able to. It won't line up with a complementary chromosome that doesn't have that huge haplotype, right? So I mean that's a big part of like the so-called crossing over, and when you mate, all your chromosomes line up, and then they exchange segments. This helps increase genetic variation within a population. It helps make sure that you don't look terribly. You're not like terribly much like either one of your parents exactly. So you can be a really healthy mix of the two.
Yeah, I didn't really know this.
Yeah,
humans don't have that many haplogroups, do they?
Well, I mean, so a haplotype is any large inherited subset, but as far as these, diseased haplotypes, not that I know of. But anyway, in the T haplotype in Mus muscus, or the house mouse, the way it works is if you have only one copy of it, gametes that have that one copy will survive. Gametes that don't have that copy will die before you can mate. So you'll only produce sex cells that have that copy. And then, that's basically the way it forces its inheritance.
That's interesting. I didn't quite know that.
Yeah.
Yeah. So the thinking is with this, I think this mouse exists too, from what I was reading. But the mouse actually is bit by a tick, and the tick uptakes the gene, the blood or something, and then it makes Lyme disease eventually work its way out of an environment. So there is like an island they want to release these mice on, or these rats. And the goal is to eliminate Lyme disease from the island.
Oh, wow.
But then people
That's
are worried
crazy.
that you've opened up a can of worms by the genetic insert. But I mean, to get rid of Lyme disease and malaria just seem seem kind of worth the risk, generally speaking, if we do it right and very carefully.
So the problem with that genetic drive approach is that you wouldn't do it to a species that you're not willing to get rid of.
Right.
So in the case of the mosquitoes, it sounds great because you're just going to lose a bunch of mosquitoes.
Yeah.
You know, what happens if we wiped out the whole species? I mean, that was kind of the goal.
They don't really serve anything.
A lot of stuff.
Yeah,
they're not
them.
important to the
Bats,
chain
other
of life.
bugs.
But the butterfly effect.
I mean,
and everything.
things eat them. I mean, that's a problem.
But
There's a whole video online of what would happen if we didn't have any mosquitoes.
really,
Yeah, and it explains all the ramifications and all the issues that it would cause.
have you heard people just since humans became like the Anthropocene, since we became like kind of impacting the 6th great extinction?
Yeah.
Because so many species have already died since humans got
Anthropocene.
kind of a good amount of technology, that it's on par with like some of the big extinctions of the past.
Oh.
But how worrisome is that? Like, this one's not because of an environmental change. I don't know. It's just weird. We're just killing so many species, but
We're just
I don't
speeding
really
up
know
that
how to just,
natural
go
cycle.
about. But I want all the things that technology brings me, so I'm guilty of it. But just maybe we could think about it a little more as a society and put rules in place. Well, that's it. Thank you so much for sharing all of these insights.
Thanks for having me. This was awesome.